One, let's understand vectors today. What are cloning vectors and what are expression vectors? But before understanding vectors, we should know what gene is. So, gene is a segment of DNA that contains instructions for making a protein. We say we resemble our parents so much because we have got those genes from our parents. So, the genes provide the information needed to make protein that performs specific functions in cells or express physical traits such as hair color or eye color. So whenever I say I have the same eye color with matching with my parent, it means I got that gene from my parent. Now you might know about one hormone, which is our insulin hormone. So it is a peptide hormone, or I can say it is a protein. So in our body there might be some gene which codes for this insulin, obviously. So this insulin, it regulates the blood glucose level in our body. When I say it regulates the blood glucose level in our blood, I mean it converts the excess glucose which is present in our body into glycogen. So it causes the cells in the liver, muscle and fat tissues to take up glucose from blood and convert it to glycogen that can be stored in liver and muscles. It is produced by beta cells of pancreas. But you might have heard about the disease named diabetes mellitus. It is one of the common disease we often hear. Most of our known might have it. So in case of diabetes mellitus, the level of glucose in the blood increases. That is, this glucose cannot be converted into glycogen. And then these people need to be given external insulin in some cases. But when I talk about external insulin, and I'm telling like insulin cases are so much nowadays, we might need a lot of insulin. So for that case, we might need a multiple copies of this gene, or we might need a large amount of this protein, right? So as I want this gene, it can't be produced in a large amount in human. Even if I talk about slaughtered animals, if we can produce them, but there will be some toxicity which can be harmful to the person where it is given. So we need its large production. We can use microbes. Talking about microbes, bacteria, yeast, we know they have a very high generation time, like they can double within minutes. In 24 hours we get a large amount of that bacteria. So we will need that bacteria, we can use it and insert this gene into them so that we can get a high amount of that gene or protein within 24 hours. But taking that gene from a human and inserting it into a bacteria might not be that easy. It is not like taking it up and putting it into bacteria and it will start multiplying. We need a thing for it. We need a transporter for it. And here comes the role of our vector. So it's like I want to go to Mumbai, so there should be someone who picks me up from here and drops me in Mumbai. So that's the work this transporter, this vector does. Also what it does is it helps this gene to replicate in that E. coli, because I told it can't be going directly into the bacteria, start replicating it. My bacteria might kill it, so there might be something which is helping it to adjust, and that adjusting power is given by this transporter known as this vector. A very good example of this vector is a plasmid. We know most of the bacteria have this plasmid. Plasmid is an extra chromosomal material which can self-replicate. Remember when I'm telling it can self-replicate? It can self-replicate because it has origin of replication. Origin of replication is a point from where the replication starts. Whenever we talk about DNA replication, we talk about this ori, which is origin of replication. So these vectors have this, and that's why they are unique, that they can be used as a transporter. So first let's understand what these vectors are. So these vectors are the DNA molecules that act as transporting vehicle which carries target DNA into a host cell for the purpose of cloning and expression. As I told you, I wanted that human gene insulin to be expressed in a bacteria. So the transporter here was the vector. But vector should have some characteristics. That's why it helps this gene to replicate as well as how will we knowing that this gene is actually inserted into the host, that is in the bacteria. So the characteristics which it has the ability to replicate in host cells because it has ori, which is origin of replication. Secondly, it has unique restriction site for insertional cloning. This unique restriction sites help us to insert our gene of interest in that specific point. Also, it has a genetic marker to select for host cells containing the vector, which means when this our gene of interest goes into the host, how will know it actually got transformed or not? So for that we need these selection markers. I will be giving a very good example. For example, a plasmid. Plasmid, if I talk about PUC, it has ampicillin resistance. So normally when that bacteria will be grown in ampicillin. Ampicillin is an antibiotic. So whenever that bacteria which contains a plasmid of ampicillin resistance, it will be grown in ampicillin containing media, it will grow. Why? Because it has that ampicillin resistance. But if I insert my gene of interest into that ampicillin resistance site, now what will happen is that bacteria will not be able to grow in the presence of ampicillin because it lost its ability. Same thing happens. So with these examples, we can check whether our gene of interest got transformed into the host or not. And lastly, it should have a low molecular weight. Now, see, we have this gene of interest. For example, I told you we need an insulin protein. So this might be a gene which is coding for insulin. And I want it to take into this host, which is a bacterial cell. So we'll be using this vector. Now, it's not like, joining these both and with some glue. We don't have any glue right now. We can't use Fevicol to just join them. So what we need to do is we need to cut these both with some restriction endonucleases. These are molecular scissors which help us to cut the DNA at some specific sequences. So we cut with the same restriction endonuclease, these both, and then we join them with the help of DNA ligase. So when we join these both, we get a recombinant DNA, and this then recombinant DNA is then put it into the host. So this vector, it acts as a transporter. It takes our gene of interest and puts it into the host where we wanted it to get multiplied. So this you can see, we had a DNA fragment, we put it into the plasmid, we got a recombinant plasmid, and then we transformed it into a host, which was a bacteria. Now, as the bacteria will double itself, these genes or those proteins will also double themselves, and then we'll get a multiple copies of these gene or protein. Now, I have been saying you we might need a multiple copies of genes or we might need a large amount of protein. But in both the cases, we need different kind of a vector. When I'm talking about we need a multiple copies of gene, we need a cloning vector. So cloning vector is the one which is used to acquire multiple copies of foreign DNA fragment, which was our gene of interest. So for this, we need in our vector our origin of replication. I told you the point from where the replication will start, our multiple cloning sites, and obviously a selectable marker. I'm sorry, we are not able to see it, but it was written here. But when I was talking about I need a large amount of proteins, we can't use a cloning vector. Proteins is a gene product. When we need a product of this gene, we need expression vector. So expression vector is a modified form of a cloning vector. So it will have an origin of replication, it will have multiple cloning sites, it will have a selectable marker, but it also have regulatory sequences. So what are these regulatory sequences? You might have heard about central dogma. In central dogma, we convert DNA is transcribed into RNA and RNA gets translated into a protein. So here comes the process of transcription and translation. And whenever we talk about transcription and translation, we need some promoter sites from where these processes starts. We need some termination sites where these processes end. So these promoter and terminator sites should be present in expression vector. Then only we'll be able to get a protein. So the major difference is expression vectors also have those promoter and terminator sequences. When I talk about the example of cloning vector, here comes the plasmid, cosmid. So we are having a lot of examples, and the major difference in these will be, like if I talk about the plasmid, the insert size. Insert size means the gene of interest I was having can be of like less than 10 kV. If I talk about cosmid, it can be less than 45 kV. So depending upon the size of my gene of interest, I will be using a particular vector. Also, they have different copy number. When I'm talking about copy number, it means how many copies of gene I will be getting. If I talk about example of expression vector, here comes PET vector, which is plasmid expression vector, or adenoviral vector. So mostly in expression vectors, we use viral vectors. So this is the example of a cloning vector, and in this it's just, I've told you, like when I was telling about the gene of interest and a vector, they need to be cut with the same restriction endonuclease. It's done so because we need same kind of sticky or blunt ends, and then they can be joined with the help of a DNA ligase. Now this is a cloning vector and other is the expression vector. If you see, there will be very much similarities. Like in cloning vector, you can see ampicillin resistance, which can be told like selectable marker, or we have origin of replication, and then we have these multiple cloning sites. In expression vector also, we can see all these three things, but here we also have promoter sequence and a terminator sequence, which makes it different from a cloning vector. Now here comes examples of vectors and insert size. As I told you, depending on the insert size, we use different kind of a vector. So here is the rough idea how much size of insert should be there to insert into a vector. So when we are using a plasmid, our insert size should be less than 10 kb. While using bacteriophage, it should be 9 to 15 kb. Cosmid should have 23 to 45 kb. BAC, which is bacterial artificial chromosome, should have less than 300 kb, and YAC, which is yeast artificial chromosome, should have insert size of 100 to 3000 kb. And one more is there, which is mammalian artificial chromosome, which can have a size larger than 3000 kb. So that's all for today. Thank you. If you like my video, do like and share my channel. Thank you.